Hey everyone, we'll just get started with the seminar. Thank you all for coming. It is my pleasure to introduce Dr. Erin. She will be giving us a lecture, a quick seminar on a topic called Fixed in Time. She's a postdoctoral research fellow from CSIRO. Now with the really good stuff. Erin has an actual formula to go back in time. She developed these new methods wherein you can extract DNA from formalin fixed collections and specimens. Think about all of the museums that we have in the world, right? And think about all of the specimens that are just there floating in a jar, right? It is not just about knowing what they were and what they are and when they were collected. It's about being able to unlock the secrets that those populations had in the past. Erin can do that. And it's something that she's been applying for a while, something that I'm very interested in working her with as well. And I'm very keen on seeing what she's going to share with us today. So do sit back, enjoy the presentation, and please welcome Erin.
Thanks, Alejandro. Thank you, everyone, for letting me talk to you today, because this is the first time that we're getting to present this body of work in its entirety. So I'm hoping that you're going to be as excited about it as we are by the end of my talk. Before I go any further, first, I would like to acknowledge that we're standing on Ngunnawal land. I would like to acknowledge them as the traditional custodians and caretakers of the lands upon which we work, live, and play. I would also like to extend that acknowledgement to any of the traditional custodians that the people who are joining us online are seated on today, and welcome and acknowledge any Aboriginal or Torres Strait Islanders people that we have with us today. This talk is going to talk about time. Before we dig in too much, I want to acknowledge that the concept of time is, of course, different depending on who you're talking to. These museum specimens that we're going to be getting data from are only collected over the last 200 years. And the insights that we can gain from them barely scratch the surface of the deep understanding that Aboriginal people have of our lands. I'm started with a depressing slide and give you some depressing statistics. It shouldn't be news to anyone in this room that we're experiencing a global biodiversity crisis. Global wildlife populations are crashing. We're experiencing an increasing rate of extinctions. And for those species that we are able to identify as needing our help, we don't have a particularly good track record of pulling them out of endangerment. Of course, it's a multifactorial problem, and there isn't any one way to solve it. But I feel that a way that we can do better is to better identify those species that are most at risk, where we can concentrate our efforts. To do this, we need to be able to understand how species are coping with their environment today, but also predict how they're going to respond to future change, because this rate of change isn't stopping. And then we need to find out how to support them and mitigate any kind of risks that they're experiencing. This is to just understand how species respond to this change. And we can do that by looking back in time at how they've responded to previous events. And then that will give us information that we can then predict how they might respond to events in the future. There's multiple ways to go about this. And I imagine most of you are familiar with looking at how species adapt to their environment through the lens of genetics. This talk is going to touch on epigenetics a few times. So I want to just kind of show you how these two work in concert together. So here's this hypothetical population of frogs. They all have slightly different phenotypes that are the result of their underlying genotype. They're all happy living in their little pond, and then they're presented with some kind of environmental threat. On the other end of that threat, you have one phenotype that was able to survive this threat. They go on and they have lots of offspring. And so this we understand as evolution of differential selection on genetic variation. I'm going to twist that a little bit. So let's say we have that same population. They're all a little bit different, all have slightly different genotypes, resulting in different phenotypes. And then you have this one individual who's able to modify its phenotype slightly through phenotypic plasticity. And it's able to, in the presence of this risk, overcome that challenge by slightly modifying its phenotype. On the revert back to its original phenotype, again through phenotypic plasticity, and then go on, have lots of little froggy babies who are also have that phenotypic plasticity that they might be able to use to overcome future threats. So there's two ways, both genetics with the help of epigenetics to modify their phenotype, that populations can persist. So this term epigenetic, people use it in different ways, and there's lots of different mechanisms that we can study in order to understand the underlying mechanisms of phenotypic plasticity. So you're probably mostly aware of DNA methylation as something that we can either use as a marker of epigenetic changes, or that we can understand of it being the underlying cause of differential expression. Then, of course, there's histone modification, so little chemical tags that are added to the packaging mechanisms of chromatin. The actual packaging itself of the chromatin is informative, so the positioning of nucleosomes in the greater chromatin structure is informative of gene expression. Or if you want to go crazy, you can measure gene expression on its own, either through RNA-seq or through proteomics. You use different epigenetic marks to understand how populations are responding to change. And if we have information about how populations have responded in the past, we can use epigenetic marks to get information about these processes. And so this is a concept that my group is calling temporal epigenomics. So it's looking at species response through long-term data studies. When you're studying temporal epigenetics, you care about a couple of things. You care about which genes are involved. You care about how quickly they're able to change their expression through the various epigenetic mechanisms. And of course, what size effect these changes are having, which all give us a better picture of how species are responding. There's different ways to go about collecting temporal epigenetic information. You can go about setting up long-term ecological research studies, collecting lots of data, doing lots of field work. And this is one really great way to collect controlled data. Unfortunately, getting at that baseline information of what species looked like, what their epigenetic profiles looked like. And so, as Alejandro alluded to, we're looking at cracking into museums as a source of temporal epigenetic information, because sitting under our fingertips are millions of specimens that have already been collected for us, representing these long-term ecological data sets. And the only thing preventing us from doing temporal epigenomic studies on these specimens is having the methods to do it. Because we can study these specimens from the perspective of how they're responding to climate change, how they're responding to new pathogens, or how they're responding to various invasive species. And so all this information is sitting there at our fingertips. In museum collections, we have specimens that are prepared in a whole bunch of different ways, and each of them presents a different challenge to extracting DNA and, of course, getting at gene expression information. One way, sort of the gold standard way of getting epigenetic information from museum specimens, we'd be going straight to our tissue vaults, looking at those flash frozen tissues that were collected when we collected the specimen. This can be a great source of RNA. This can be a source of DNA methylation. You name it, you can probably get it out of these tissues. Unfortunately, sampling with frozen tissue only started relatively recently. And so getting at specimens that represent the state before massive change is impossible with these kind of specimens. We also have huge banks of dry tissue, so bones, hides, teeth. These can go much, much deeper in time. And there have been a few studies looking at especially DNA methylation of these sorts of tissues, but it's only telling you the information because it's tissue specific of how bones or teeth or hide are responding to environmental change. And as you can imagine, it probably doesn't tell you how this animal might have responded to a novel respiratory virus. In order to get at that tissue-specific information, we need to find specimens that are preserved whole. We have a whole bunch of those. We have hundreds of years and thousands of specimens preserved in spirit. These specimens either fixed with formalin or not, sometimes more with ethanol, sometimes unknown fixative, and then put in their vaults and preserved for hundreds of years. These would be the ideal candidate for studying tissue-specific epigenetic information, but unfortunately, there's this long-standing thought that formalin preservation itself completely destroys DNA. And that's what we're completely turning on its head. Ought to degrade DNA because of the fixation process itself. Formalin chemically cross-links all of the little proteins and everything in the cell to all of the DNA. So that in and of itself either degrades the DNA, or the processes that you use to get the DNA out of the tissue then fragment the DNA. And so most people, when they try to extract DNA out of formalin-preserved tissues, it doesn't work particularly well. Field of immunohistochemistry from medical biology, we know that there are a lot of chromatin assays, so studying that shape of the chromatin, that use formalin in order to take a snapshot in time and see what the cell was doing. So if formalin completely destroys DNA, how is it that they're able to get this really fine detailed information out of cells that they've fixed with formalin? There's a bunch of different assays that use formalin. You might be familiar with a few of them. So there's chromatin immunoprecipitation assays, which use an antibody to pull out different pieces that you're interested in. There's Hi-C, which gives us that really gross information about which parts of the genome interact with other parts. And there's two more bespoke methods called FAIRE-seq, which uses fraction the genome and everything. Whoop, what did I do? Push the button. Oh, good. So everything that is cross-linked protein, so all the DNA that is locked up tight and not being expressed goes into one part of the assay, and everything that is open and expressed is put into the aqueous phase, and you can pull out that and enrich for that. There's also micrococcal nuclease sequencing, which uses an enzyme that goes in and clips on either side of the nucleosome, chews up the DNA, and then what you're left with is that fingerprint of where nucleosomes were sitting on the DNA. Before to use those kind of complex assays on museum specimens, we first had to shake the tree and prove that you can get DNA out of formalin-preserved specimens just in and of itself. And our group went through a long process of figuring out a vetting system, first and foremost, of finding those specimens that are most likely to yield DNA. There's differential preservation practices in museums. So sometimes a specimen will take quite a long time before it gets into formalin. A lot of the DNA will have degraded just naturally due to postmortem decomposition. We want to avoid those ones. We also want to avoid specimens that have been fixed in preservatives that really do degrade DNA, so like formalin acetic acid. So we had a system that we can go in, we can take a look at the specimen, we can take little sips of the media, and then guide ourselves towards those specimens that are going to be most useful. We then put it through a process of DNA extraction, which is modified to contend with that really heavy fixation. And I think the biggest part that we do differently is our bioinformatics, where we're really, really squeezing as much as we possibly can out of this highly degraded DNA using a program called K-Align, which fortunately for us is also really easy to use because it doesn't require any trimming or any kind of QC filtering. You just slap it in and hopefully it comes out of it. And it comes out with really. We published this work in Molecular Ecology Resources last year. And at that time, we were getting about 3 or 4x cover from some of our better specimens. And since publishing that, we've also had really, really good success with really old specimens, getting up to 11x cover on Eastern Water Dragon specimens from 1905, and also really, really small specimens. So infinitesimally small tuna larvae that we're getting upwards of 25x genomic cover from. So we've smashed that assumption that you can't get whole genomes out of formalin-preserved tissues. Next to work on those chromatin assays. We didn't try ChIP-seq because formalin can modify the epitopes that antibodies will recognize. And so that was a whole quagmire that we didn't necessarily want to get into. And we wanted something that's going to work in all species and not necessarily just be skewed towards those with well-developed antibodies. And something like Hi-C, we're also worried that that really big, like long-term structure might be destroyed by the processes that we're using to get the DNA out. So we focused on these two other methods. Collection and using really old specimens to try to work out the optimization parameters for these different assays, we first sought to understand exactly what formalin is doing to chromatin. To do this, we chose a species that's really easy to grow in the lab and doesn't require any ethics approval, and you can do all sorts of treatments to it in a matter of a day. And so for this, we chose to use Saccharomyces cerevisiae to expose it to an environmental condition, in this case, one that's really well characterized, putting it through heat shock, and then fixing it for different periods of time, 15 minutes, which is kind of the standard for these types of assays, up to 24 hours, which is like way, way, way overkill for fixing yeast. Then we put it through these two assays to see what happened to the chromatin of DNA sequence alignments from heat-shocked Saccharomyces cerevisiae just upstream of a gene that's turned on in response to heat shock. So we'd expect this gene to be open, accessible, and highly expressed. And this is a picture of what MNase looks like when you perform that on these different cells that fixate at different fixation points. So this is at 15 minutes, this is a standard MNase profile. You see these little humps where the nucleosomes are sitting. As we increase fixation time from 15 minutes down to 24 hours, we see that signal change. Rather than seeing these undulating waves of nucleosomes, we see this big honking peak right here. Is under short-term fixation, the nucleosomes are snipped, and you get this enrichment right here where all the nucleosomes are sitting. That's because the chromatin itself isn't as tightly wadded up into essentially what amounts to like cement. When you get really, really heavy fixation, formalin's allowed to go crazy. Everything cross-links to everything else. And it's only those most open, accessible bits of the chromatin where you're even going to be able to get the micrococcus enzyme in to do the digestion. And so in that case, it's just the more open parts of the chromatin where you're able to digest and get enrichment. And so these undulating peaks turn into one singular peak. So now what happens under FAIRE treatment? This is what you generally see when you treat FAIRE after standard fixation. For 15 minutes, you just see a peak at the very beginning of the genes. That's big open parts of the chromatin that is being enriched. Here, as fixation time increases, that signal changes. And this isn't random right here. This is a signal that we're seeing upstream of a lot of genes. Under short fixation, that open part of the gene, part of the chromatin is enriched for, and you get a peak. And what we think is happening when you apply FAIRE to really heavily fixed samples is that in these open regions, it isn't just nucleosomes. Inside the cell, there's lots of other different parts of the cellular machinery, the transcriptional machinery that are interacting with the DNA at different times. And when formalin's allowed to go crazy for a long period of time, you're fixing more of that protein onto the DNA, in particular, RNA polymerase and the transcriptional machinery at open sites. And so what we think we're seeing here upstream of genes is enrichment of the overall open region, but then depletion where RNA polymerase is sitting. And we see this consistently across the genome of the signal of peak with a little apical dimple on top. To what we're able to do with this kind of data, again, looking at it in terms of what's our data from Saccharomyces cerevisiae, we took advantage of having run this experimental environmental change process to the yeast, so in response to heat shock. So it took us a little while to figure out how to analyze these data, because as you can see from this slide here, it isn't just enrichment. Sometimes it's that nuance of depletion that we're looking for. So we're looking for where you have enriched signal as well as loss. And there aren't a lot of peak calling programs that do that. Most of them are just looking for where you get big seeks, like big sequencing peaks, like you would see in ChIP-seq. So we found this program called DANPOS, which looks for variation in sequence alignment, both in terms of enrichment and depletion, and gives it all to you in one nice little package, which is really convenient. When we apply the DANPOS model, so looking at differences in signal between heat shock and the control samples, what we see is this is what we have a signal of differential gene expression. Under normal fixation conditions, upstream of a very highly upregulated gene, which here is a heat shock protein, this is what differential peakiness looks like upstream in an MNase assay. So you have different heights of those peaks, those undulating peaks. And DANPOS goes in and it says, this is where they're significantly different. So in MNase, you have a significantly enriched region upstream of a heat shock protein. In FAIRE, the distance of that open region is variable, but what you see again is enrichment. In our heavily fixed yeast, however, that signal changes. We already know kind of what is changing between the two sample types. And now if you apply a differential analysis to these data, what you see is a noisier, but still signal of enriched read depth in heavy fixation in the MNase assay. And here we see that DAMPOS has not picked up any significant difference between the two cultures in heavily fixed FAIRE. So we're starting to see an inkling in the difference in sensitivity between these two assays. On the flip side of that, we can look for differential expression signals at a gene that's very heavily downregulated. So what happens when we have the opposite between the two cultures? So here at this downregulated gene, an MNase signal, although there's a little bit of enrichment, and that's because there's a lot of different peaks here, DAMPOS is picking up a predominantly significant decline in sequence reads in the heat shock protein relative to control. So we see depletion signal at these downregulated genes. Our FAIRE treatment didn't do so well at a regular time point in order to pick up a depletion signal, and that's because FAIRE does a lot better at picking up genes which have a strong on signal. When you look at the heavily fixed tissues, however, our heavily fixed micrococcal nuclease treated samples, again, show you a very heavily depleted signal at this downregulated gene. And if you look at the order of the signal change here, this is an order of magnitude stronger than in the fresh tissues. So they're still picking up a negative signal, a depletion, but it's much, much stronger in heavily fixed tissues. In our FAIRE assay, here we have a signal of a depletion upstream of this gene. And so even though the fresh sample didn't pick up depletion, FAIRE did. And so there's something that is added, additional information that is added because of the fixation, rather than eliminating the signal entirely. So that's all well and good to be able to see sequence reads upstream of genes. How does this actually relate to us understanding the biology of these species a little bit better? So ideally, we get gene expression information out of these yeasts. And I was able to do that with a fresh sample, but not with the heavily fixed samples. So this is looking at trying to use that signal strength itself in order to see if there's any information lying in that to give us information about gene expression. Because I mentioned here in these depletion regions, a fresh sample has a signal that's fairly low, but in a depletion sample here with heavily fixation, it's really, really low. So something else is happening here. When I did an analysis of the correlation between gene expression through RNA-seq on the x-axis and total signal height on the y-axis, under normal fixation conditions, there isn't any signal. It's either up or down, and it doesn't really matter how high it is. Under heavy fixation, however, there is a correlation where those genes that are highly expressed have a high signal change, and those genes that are lowly expressed have a lower signal. So this gives us that inclination that we can then infer gene expression information from heavy fixation, which you can't do with normal assays. So not only can we apply these assays to heavily fixed cells, we get extra information. Going one step further, can we infer more broad scale information from a biological perspective from these data? With RNA-seq, you sometimes do a Go analysis where you're trying to figure out which biological processes or pathways are enriched in your genes that you're seeing as differentially expressed. So again, this is looking at differential expression between heat shock and control yeast. RNA-seq is telling us that those genes that are upregulated are enriched for these pathways, which all have to do with the heat shock. That's good. When we look at the archival micrococcal nuclease signal gain, we have similar pathways being enriched with this assay as to RNA-seq. So we're seeing parallel information through archival samples. And so MNase did that. FAIRE didn't do that so well with the upregulated genes. However, when we look at those genes that are downregulated, where we're seeing signal loss for FAIRE, we see a parallel type grouping of biological processes enriched in both pathways. And so while FAIRE is great at finding those genes that are highly upregulated, here it's giving a good signal of those which are downregulated. And so this would give us sort of two ways of tackling this beast to get different ends of the equation. Okay, so we've got this figured out in yeast. That's all well and good. We actually want to apply this to museum specimens. There's a couple other hurdles that we need to overcome in order to start applying this to multicellular and multi-tissue vertebrates. To do this, instead of using precious, irreplaceable museum specimens, we set up an experimental specimen set a number of years ago. We selected to do this in mice because they have a really well-characterized genome, and because you can access inbred strains so that you're working in a system where there isn't as much genetic variation. And then we also wanted to include a group of wild-caught mice in order to start infusing that with genetic variation to see how that influences the signal that we're getting. So we created this bank of specimens by taking fresh liver tissue. So each of these has a matched fresh sample that we can compare our archival signal to. Then we formalin preserved them, just like we do our museum collection specimens, and put them in the vault for between 2.3 and 4.8 years. So these are properly fixed specimens that are done in a way that we normally do. Of course, we just know exactly how we did it. And then we put them through our fresh assays as well as our archival assays. The archival assays took a bit of work to get them to be optimized to vertebrate tissues. And this component of the assay here, where we're just going from tissue itself to trying to isolate nuclei, took about 5 years of work. And it's because it's like chiseling a very fine strand of hair out of cement. You both want to use a jackhammer and you want to use a very fine brush to get at it. And so this is a whole bunch of combination of different methods of intermittently going hard and then very carefully teasing the chromatin out of the heavily fixed tissue. It involves a whole long process of cryopulverizing the tissue, putting it through a pepsin digestion, which is just a gross protein digestion, just to kind of go in and clip here and there. And then putting it through initial decross-linking step, as well as downsinging to get that tissue much more finely ground. Once it's a finely ground, slightly digested, slightly decross-linked state, we put it through a sonication process, which isolates nuclei from the whole cellular matrix and all the junk in the cell. So that was quite a lot of work. But once we then started focusing on the assays themselves, there were two different parts that we needed to optimize with the two assays. For FAIRE, optimization primarily focused around the shearing conditions that we put it through in order to chop up the DNA. It's counterintuitive to think that you need to chop up fragmented DNA, but we found that we actually needed to do that in order to get any DNA out of it at all. And then we're putting it through a phenylchloroform extraction where the locked up tight stuff is meant to go into the organic phase and the open stuff is meant to go into the aqueous phase. With the micrococcal nuclease assay, it was mostly optimization around digestion time. So how long you're leaving the enzyme in with the tissue and how much of the enzyme you're using. Once we were satisfied that we're getting DNA out of it, doesn't look like the DNA profile you would get from fresh assays, but once we were confident that we were getting enough for sequencing that was going to give us nice, rich libraries, we put it through a library prep protocol that we had been using for our highly degraded genomic sequencing assay, which involves an IDT library prep, put it in through very, very heavy sequencing. And then the analysis is, again, similar to what we do with our whole genome sequence alignments. We put it through K-align. We use a GC bias correction to make sure that we're not enriching for sequence that naturally just sequences better. And then we put it through our DAM post-enrichment analysis. What like when you put heavily fixed tissue through these assays? Just to give you an idea of what a fresh tissue sample is supposed to look like. This is data from, again, our inbred lab mice to start with. Our MNase of a gene that is highly expressed in the liver, this is the tissue that we sampled. What you get from a fresh signal isn't exactly those undulating signals. It's a little bit noisier with some an undulating signal here. And DAM post picks up a predominant signal of enrichment. As in FAIRE, DAM post picks up a predominant signal of enrichment upstream. The signal looks different, again, like it did in yeast, but different again. In archival treated mouse samples treated with MNase, Rather than seeing enrichment upstream of a gene expressed in liver, what we see is strong depletion, sort of a mirror image. And likewise, we see kind of the same thing in FAIRE. In our archival tissues, we see a very strong depletion at genes upstream of genes that are expressed. This is what we think is happening here. So just like in the yeast cells, under short fixation, you get little clips on either side of the nucleosome, you get enrichment. It's a bit noisier because these are multicellular organisms, they're not clonal. Under heavy fixation, you still have that whole quagmire of chromatin that you're trying to extract. I think that most of this chromatin that is really, really heavily bound and unexpressed isn't accessible to the micrococcal nuclease. So instead of in yeast, where you're just clipping on either side and isolating that nucleosome, micrococcal nuclease, if you give it half a chance, will digest everything. And so what it's doing is it's digesting those most open regions, leaving you with a valley where you would normally have seen a peak in right? You're seeing that mirror image here. In the FAIRE assay, it's a different process that I think is degrading the sample there and causing depletion, but it leaves the similar kind of signal. So FAIRE, again, you're going to see an open peak when you're using fresh tissues. Under long-term fixation, for us to get any DNA out of it, you have to be quite vigorous. And this involved a heavier sonication protocol in order to release any DNA. And what I think what is happening is, in conjunction with just some natural degradation that we're seeing in museum specimens, heavy sonication just obliterates all those sequences that are open and accessible, leaving some of that more bound up stuff, which itself is sonicated and released. You sequence that and you're getting depletion of these most open regions. And so you see a similar signal, but for a different reason. So these are complementary assays to get us at the same kind of information. And to show you that this isn't, again, just random, this is upstream of a gene that is not expressed in liver. So we can see that neither the fresh assays or the archival assays pick up any signal of there being anything happening at this particular gene. It's not on. This also is a phenomenon that we see across the genome. I've shown you a couple genes that are indicative of what's happening at a gene that is expressed in the liver, but what's happening across the rest of the genome. So again, from our lab mice, we're looking at how this signal compares at the transcription start site right here, all lined up, all the genes across the genome. We see this tornado-like effect, and that's showing enrichment of peak gain, so that signal enrichment that is sitting right at the transcription start site, which we'd expect for genes which are active and open. And we see that in both assays. And this is what a proper MNase or FAIRE profile is meant to look like across the genome. When you move to archival tissues, rather than looking for peak gain like you do in fresh tissues, we flip that and look for peak loss. And when we look for peak loss through the tissues that are treated with MNase and FAIRE, we see that same kind of tornado effect where there's enrichment at the transcription start site all across the genome. MNase looks more or less like it does with in fresh tissues, and FAIRE, the signal's weaker, but it's still there. Now, can we get information about gene expression from these? Because that's ultimately what we want. And here we took advantage of having sampled two different populations. So, you know, ideally these have undergone different environments. These were out in the bush and these ones were in a lab. Did RNA-seq analysis to assess how the genes were expressed differently in the fresh tissue. And then we looked at comparing the signals from the fresh and archival MNase and FAIRE treatments between these individuals to see how those signals change. In our fresh tissue, treated with micrococcal nuclease, this is a gene that is like most differentially expressed between these two populations. Upstream of that gene, MNase gives you a nice positive enrichment that the lab mice are enriched relative to our wild mice. Likewise with FAIRE, we can see through our fresh tissue, we see a nice positive enrichment signal. So yay, it's indicative of gene expression. When we then treat the archival samples, rather than seeing that positive signal, we see Dampos picking up a significant depletion signal. So we have a signal. It's not what we would see with fresh tissues. We just have to think about it a little bit differently, analyze it a little bit differently, but we can pull out that similar information if we understand to expect a signal loss. We got it to work in mice, lab mice. That's great. We did some intentional fixation under conditions that we knew were going to work really well for our types of assays. We want to put it to work on actual museum specimens where we never met the curators, we didn't see it happen, we don't know what's going on. We teamed up with our collaborators up at UQ, Celine FAIRE and her PhD student, Nicola. They study eastern water dragons, and they're primarily interested in understanding how this species has adapted to the rapid urbanization happening in the Brisbane area. They have a really wonderful modern data set, which includes not just genomics, but behavioral information, changes in their morphology, and changes in the susceptibility to a fungal disease that is hitting the populations pretty hard. And so they have a really good idea about how the modern genetic differentiation and these various traits are sitting across the landscape. They really wanted information on what Brisbane area looked like before all of this urbanization and during. To do that, they needed historical specimens. And because most reptiles are fixed with formalin, that was a big barrier for them. And they came to us and asked, can you get genetic information out of our water dragons? And we said, yeah, we're pretty sure we can. Would you let us try this other crazy assay on them as well? So we sent Nicola into the Queensland Museum with our specimen vetting protocol in hand. She found a bunch of specimens that we thought were suitable for sequencing, as well as a number of specimens where she was able to get enough tissue for us to really try hard to use both our genomic sequencing assay and one of our archival chromatin assays on. We chose one because at that time we hadn't quite figured out how much tissue we needed. And so we wanted to put all of our effort into the one that seemed to be giving us more signal. And so that was the archival micrococcal nuclease. The 5 specimens that we used for this trial, 4 were male, 1 was female, and the oldest one was from 1905. And then we have a few from the 1990s and the early 2000s. Jumping right into it, what does archival micrococcal nuclease sequence data look like across the genome of truly archival, properly old eastern water dragons? So these are those similar profiles of enrichment at the transcription start site, again, a peak loss in these 5 dragons. And what you see is an enrichment, some stronger than others, right at the transcription start site. And what's really interesting is that these 2, the more recent samples, don't look all that much different from the oldest sample. And so time in and of itself isn't the determining factor of whether or not you can get information out of these specimens. It's more likely how much time was in between collecting the individual and popping it into formalin. As a side note, just showing what kind of whole genome sequencing cover we're getting out of these specimens. If we combined our whole genome sequencing data and our micrococcal nuclease data, we're getting upwards of a 14x cover of the whole genome. So this is high enough to call SNPs, which we're doing once our bioinformatician gets back from leave. So in this assay, we're able to get whole genome sequencing and archival gene expression information all in one go, which is super, super exciting. Of course, it's just 5 specimens, and they were collected at different points in time, at different geographic locations. We have a whole bunch of different things going on. So we couldn't really dig into, say, the effect of fungal pathogens on these 5 specimens. But we decided to chuck it into analysis looking at the correlation between the profiles, the MNase profiles between individuals. And something quite striking jumped out at us. Here we have our 5 samples. And so the color just indicates how similar or how different they are from one another. And we have one individual that stands out from the others. And that happens to be a female, which to us is a strong indicator of sex differentiation in the micrococcal nuclease profiles. Wouldn't that be great if we had more samples in order to test that? Thankfully, we did. In our mouse samples, we ran that same analysis. We're just looking grossly at the profile that we get from micrococcal nuclease in our archival samples. And we see that combining all 6 of our samples that we processed, 2 of them were female, 4 of them were male. The 2 females stand out on their own. The 3 lab mice form a cluster that then has a sister group of the 1 wild mouse that was a male. Again, a small sample set, but we're starting to see sort of like inklings of the biology of the phenotype being replicated by these assays. And some folks that I've talked to who do DNA methylation say that you need to be very careful to sex match your individuals, because the signal that you get, if you're getting true epigenetic signals from your data, the first one you get is sex. And you need to match for that if you're going to pull out something more meaningful. And so this tells us that we're on the right path. We just need to sample more individuals and make sure that we're carefully matching them in order to do proper controls. And that brings us to the next phase of our project. We're pretty happy with how our methods are coming along. We have a pathway to get in, find the right specimens, process them appropriately, analyze them, and pull out meaningful data. And so the next phase of our project is going to be to form a foundational data set that researchers like yourself can then use to compare historical epigenotypes in this context of better understanding in a much greater number of taxa. So that's going to involve making the methods much more high throughput. It is a highly repetitive carpal tunnel inducing protocol at the moment, but we're going to make it something that's much more high throughput, and then apply that to 100 epigenomes from 100 different species across time so that we can get this really deep understanding of temporal epigenomics from different taxonomic groups. And so we are really excited to start down that path. So, like I said, this has been a work in progress. This is something that was the brainchild of Claire Holloway 7 or 8 years ago now, and has been contributed to by a number of researchers across CSIRO and UQ, in particular, Marina Alexander and Alicia Greeley, who started some of the optimization work and gave me a really good footing to take it off and finish off with my postdoc. And our bioinformatician, Yuri Stiller, who without him, we would never have gotten through terabytes of data in order to pull these signals out. And of course, our partners up at UQ. This work was funded by the Epigenomics Future Science Platform, which funded the work as well as my postdoc. We got sequencing funds from Bioplatform Australia, and AGRF has been really, really helpful in getting us to find those right library preps and right sequencing depth for our samples. With that, questions in the audience.
Thank you, Erin. I was thinking about insects, right? And then about what you showed with the RNA-seq and the mRNA's outcomes from gene expression. With those collections where we have bugs in the jars, is this something that could be applicable to that as well? And thinking about finding those insects that are commonly accepted as bioindicators and maybe doing something like gene expression for like for example, heat resistant tolerant genes, and is this something that could be applicable in that regard?
I'm assuming you mean individual insects,
yes. Yeah.
Rather than bulk sequencing. I think so, and I think so for a couple of reasons. One, when we initially started doing this assay, we needed like 50 milligrams of tissue, and we've got that down to requiring much, much, much less tissue. Like with our tuna larvae, we're getting super high coverage from itty bitty specimens. So it's just a matter of optimizing it down. We didn't need to do that with dragons, they're big, but we can do that with insects. Insects, whether or not they methylate or what they do, I don't know. But if we're looking at that next level of that gross chromatin structure, we might be able to get there. And if you're looking for things that are happening like on a population level, maybe just pooling a whole bunch of individuals into one, you don't need to worry about individual sequencing individuals. I think that you could do that so long as we have a basic understanding of what insect chromosomes look like, which I wouldn't be the person to ask. Yeah.
Well, it was really good talk and really exciting work. So I'm sure it's going to be transformational in understanding the past in order to better understand the future. So great stuff. I've got sort of one short question, one longer one. AttackSeq, how is that connected through this in the context? Why have you used FAIRE as the foundation and not AttackSeq?
Yeah, so the easy answer is we had to pick something. And it's FAIRE initially because we'd heard that generally the problem with FAIRE not giving enough information is underfixing it. And so going over the deep end into heavy fixation might give you more information, which is what we're starting to see. And I know that a number of years ago, AttackSeq was generally being done on fresh tissue, but now it's starting to be done in FFPE. And so I think it was a matter of us just getting stuck into some methods and going with them.
Yeah, just a timing thing.
I don't see any reason why you can't try it. And I think it would be great to then also get the parallel data sets, because no one's doing FAIRE sequencing, but lots of people have AttackSeq data for modern specimens. And if you treat the samples appropriately, get it down to nucleus, to the individual nuclei, I don't see any reason why you can't.
Okay.
The transposon insertion was going to work in a heavily fixed situation. So it was sort of bringing another variable into the mix. So these methods relied purely on digestion. But when you have to have a transposon insertion for it to work, we were like, oh, let's not start there.
Okay,
Yeah, and like
fair enough.
with the micrococcal nuclease, it's a matter of getting the enzyme in to do the digestion. It might be that the transposon insertion is just bigger and clunkier,
Yeah.
and you might get anything.
Fair
But
enough. And I guess the second question relates to the fact that gene expression is like cell line dependent. And so the first thing is that means you have to pick the same organ, but there's also the possibility that there are changes in cellular composition in those organs that will add noise to any trend you might be looking through in time, and also the individual variability in relation to time of season, all that sort of stuff. So how dominant do you think that noise component is going to be in sort of comparing species across the 100 years? And do you think the bulk sequencing approach is going to accommodate that potential problem?
no, it's a really good question. And that's one that we're grappling with now, is how to advise on sampling numbers and exactly like how well do you need to match everything and control for as many things as you can. And I think that the short answer is we understand there's noise in fresh tissues. We know also that genomic data from archival samples is inherently noisier. When you're adding more noise due to the sequencing method and then a temporal component, it's likely that we're just going to have to get more individuals and accept that it's going to be a bit lighter touch in terms of our understanding of the individual genes. It might be that we understand which pathways are enriched. I think it kind of depends on how much effort you want to go into sequencing, into how deeply we're going to understand it. But yes, it's definitely going to be noisier data. And we have to, as much as we can, understand the age, the sex, and match them as best we can. With collection times, it's often a researcher going out into the field and grabbing a bunch that they can in a single sampling. And then you have replicates at a single sampling site. And researchers tend to go back to the same place and then put them in the museum.
Larger examination. So
Yeah, exactly. And I've been thinking about how to do a reduced representation targeting assay. Like maybe it comes at the end of the day, if you know what gene it is, a qPCR approach would help get you 500 individuals instead of 5 whole epigenomes. Yeah.
Any other questions in the audience? Chat as well. No one has any burning questions for Erin?
Well, if anybody's too shy, I've got my email, I've got Twitter here, and we have a website set up if you need anything to jog your memory.
Cool. Please join me in thanking Erin for a very interesting talk.